And good evening, everyone. So what I wanted to try and do over the next 15 or 20 minutes was just kind of introduce some basic terms and concepts that will hopefully help you enjoy some of the talks that are coming later. So I wanted to go through some of the basic concepts in genetics, introduce some of the technology that's used to help diagnose epilepsy from the genetic perspective. I was going to have one very brief slide to try and summarize what our current clinical understanding is of the genetics of epilepsy. And then finally, I was going to introduce or describe a system that we've recently got funding from Science Foundation Ireland to install at Beaumont Hospital that will hopefully help us understand epilepsy better at a genetic level in Ireland and internationally. So just to start at the start, with genetic research, the start point is this molecule DNA. And you can ask the obvious question of what DNA is. Well, it's a chemical that's contained within all, or certainly the vast majority of cells within our body. And DNA is a remarkable chemical because it's basically the book of life. And I think one of the really interesting things about that book is that we all have our own unique copy. It's referred to as the book of life because it contains the instructions for our cells to produce proteins. And that's really the main thing that our body is made up of, these many, many different proteins. So contained within this organelle called the nucleus within all of our cells, we have this chemical DNA that contains this code that tells our cells how to produce proteins. And the code is 3 billion bases long. It's made up of 4 different letters, A's, T's, C's, and G's. And if you're to read out that sequence just at the normal reading speed, A, T, C, C, G, and so on, it would take you about 30 to 40 years to read the entire code. About 10 years ago, the first sequence of a human genome, we use this term genome to refer to all of an individual's DNA collectively. So the first complete genome was sequenced by a number of different centers around the world working together. It cost hundreds of millions of dollars. There was thousands of people working on the project. And this is just an illustration of the different chapters of the book for that one particular genome. The DNA that I'm referring to, these 3 billion bases, if you look within the nucleus of the cell, you don't see that DNA as one long strand. It's arrayed across these structures that we refer to as chromosomes. So we have 22 of these particular chromosomes that we call autosomes, and then we have these two, the X and the Y, that determine our gender. Now you might notice that you've got 2 copies of each of these particular chromosomes, and that's because we inherit one from our mother and one from our father. Another term that you may have heard a lot of, whether it's watching the movies or reading newspapers or books or listening to the media, is this idea of a gene. So you can ask, well, what is a gene? A gene is simply part of our DNA that actually codes for one of those proteins. Because a lot of our DNA, something like 98%, although it contains that sequence of A, T, C's, and G's, it doesn't actually code for genes. Only about 2 to 3% actually codes for genes, which in turn code for proteins. So this is just a picture, an image of both the mother and father copy of chromosome 2 in a particular individual. And this red band is just to represent this is where the gene lies that encodes for this particular protein, SCN1A, which happens to be a very important gene for epilepsy. So there's the sequence of the gene. That then creates this particular molecule that we call RNA, and Professor Henschel is going to speak a little bit about RNA later on. And then that molecule is in turn transformed into this particular protein, which is the sodium channel. Within our genome, we have around 21,000 genes, and nature has some really kind of quirky ways to produce more proteins or more than one protein from a particular gene. So there's roughly 90,000 proteins produced by our roughly 21,000 different genes. Okay, another term that we hear a lot about is the idea of a mutation. A mutation is a change in the sequence of that genetic code. All right, so we all carry this genome with 3 billion bases of DNA, but if you compare my genome to anyone else's genome in the room, there's going to be differences between them, okay? And those differences we refer to as mutations. So these are changes in the sequence of the genetic code. So how do mutations occur? Well, they occur whenever our cells divide, right? And right now you're listening to me speaking, but within all of your bodies, there are cells constantly dividing. If the cell is dividing, say, for example, to make new skin tissue, there can be mistakes made in the production of new cells, and although our bodies have really good ways to try and identify those mistakes, sometimes they're allowed to pass through. Now, if such a mutation happens, it'll be localized to your skin, so our children don't inherit those changes. However, when we produce sperm and eggs, so these are the cells that obviously fuse to form an embryo, these changes that occur in sperm and eggs, these are changes that our children can actually inherit. And we call these changes that occur during the formation of sperm and eggs de novo mutations, or new mutations. Now, it's really important to appreciate that we all carry these mutations. All of our genomes are full of genetic variation. And it's this variation that contributes to the diversity that you see within or across any population or populations. These changes can influence our eye color, our hair color. They can influence your ability to run fast. They can sometimes influence your health. They can cause particular diseases, and they can also influence how you might respond to treatments for those diseases. But it's really important to appreciate that we all carry this genetic variation within our genomes. So we mentioned, or I discussed about where these mutations come from, where they originate from, and the mutations that we pass on to the next generation are those that are produced within the formation of sperm and eggs. And one of the things that's been realized by genetic researchers in the last couple of years is that the age of the father at conception has a remarkably large influence over the number of new mutations that are introduced. So on average, we'll all carry roughly 70 of these de novo or new mutations within our genomes. So these are mutations that our parents didn't carry, but they occurred during the production of sperm and eggs, and these are mutations that we thus have inherited. So on average, 70 mutations, but if the father is older at conception, that can introduce additional de novo mutations. So roughly for every one de novo mutation contributed by the mother, there's roughly 4 from the father. And why might that be the case? Well, the reason for that is that the age at which sperm and eggs are produced, or the sequence is produced, is very different in males and females. Males produce sperm during adult life. So the replication, the cell division that goes on to produce sperm happens in adult life. In females, on the other hand, the sequence, the genetic sequence that's contained within eggs that are released throughout life, that sequence is determined during the development of that individual in the womb. So in other words, when a female child is born, she already contains the eggs and the genetic sequence contained within those eggs that are going to be released in later life. However, when that cell division happens in the womb, the cells are just better at replicating faithfully the genetic sequence and catching any mistakes before they're passed on. Whereas in adult life, in the males, that ability to replicate the DNA exactly is lessened with age. And for that reason, more of those de novo mutations are introduced. Now, what's the significance of those mutations? Well, if you consider the genome is 3 billion bases long, right, and we said that only 2% codes for genes, anytime a mutation happens, it will happen at random anywhere across those 3 billion bases. So the chances are it's not going to make a difference. And that's why most genetic variation that exists in the human population doesn't really do anything of significance in terms of how an individual might look or their predisposition to disease and so on. However, in some cases, that mutation might land in a gene or an area that's important for that individual. And that might lead them perhaps to be better at uptaking oxygen, and they might be a good long-distance runner. Or it might happen in another gene and do something bad to that gene, where for that particular individual, it could cause some sort of a condition that requires treatment, including, in some cases, epilepsy. So with each new birth that arrives, there is new genetic variation that's introduced to the population. As we said, sometimes that can be a good thing for the individual, sometimes that has no real effect on the individual, and sometimes it can cause conditions that require treatment. If that individual grows up to have children, that variation is passed on to the next generation. And it's in this way that the human population today is full of genetic variation. This slide is just to kind of show you the types of genetic changes that we see. The only thing I want you to appreciate from this is that sometimes the changes can be very, very large and relatively easy to see when we study genes. And by large, I mean that there could be several million bases, a big chunk of DNA that's kind of deleted or duplicated within an individual. So when we study genetic sequence, those kinds of changes are relatively easy to detect. And sometimes the changes can be very, very small. So just one of those single letters of 3 billion letters might change. So those kind of changes are more difficult to detect, but today the technologies are becoming available to detect them. Okay, so for the next couple of slides, I just wanted to speak a little bit about the different technologies that are used to help diagnose epilepsy at a genetic level. And the first type we call G-banded karyotyping. And what this does is basically, it's a technique to allow you to look at the number and the structure of chromosomes under a microscope. So what we're seeing here are the different chromosomes of a particular individual, and they've kind of lined up each pair of chromosome once. So remember, one comes from your mother, one comes from your father, and so on. So they're just lined up nicely. There's been a bit of work to get this image manipulated so they'd line up like that. But by counting the number of chromosomes, sometimes you might see an individual who's got an extra chromosome. So for example, this individual has 3 copies of chromosome 21. So this would be indicative of Down syndrome in this particular patient. So with this kind of technique of karyotyping, you can literally count the number of chromosomes and make sure the number is what's expected. And sometimes you can also see major changes in a chromosome based on the banding pattern. So this is just the dark and white banding. That's characteristic within a chromosome. But if that pattern changes in an individual, that can be suggestive of a large change at a genetic level in that individual. So that technique is really good for picking up the very large changes. Another technique that's been developed more recently is called array comparative genomic hybridization, or array CGH. So this is a test that's used clinically as well. It's basically trying to do the same thing, detect very large changes in the DNA, but the range can go down to much smaller changes. So it goes down, instead of being maybe 5 million bases for the karyotyping, this one goes down to maybe 20,000 bases. So it's basically, you can think of it like a much more powerful microscope. The third technique, and I think this is one that's particularly exciting now for clinical genetics and for making a difference in the clinic, is what they're calling next generation sequencing. So with this technique, you're actually looking at the DNA on a letter by letter level. Okay, so you're not looking for very big changes of 1 million bases, you're actually looking at each individual letter. And with this technique, it's now possible to sequence or read all 3.5 or 3 billion bases of DNA for a particular individual. And I said at the start of the lecture that that was something that would have cost hundreds of millions of dollars just 10 years ago. Today, this can be done for in the region of a couple of €1,000 for a particular individual. Okay, so this allows us now to pick up those very, very small changes. And I kind of mentioned there that the cost of this technique is dropping very, very quickly. So this was when the first human genome was sequenced, so that those chapters of the book that you saw, that I showed the picture of in the Sanger Center, cost a lot of money. But then around 2008, there was a new technology developed. This is the next generation sequencing, where the price started to drop dramatically to the point where today it's down around a couple of $1,000 a year to sequence a genome. And just to put that in context, this white line here is Moore's law of processing speed. So it's basically saying that the cost of computer processing halves every 2 years. So we all have a sense of our computers getting more and more powerful with time. Well, this is showing you how the cost of sequencing is dropping far, far quicker than the cost of processor speed. And really, the take-home message for this is we now have a technique to sequence all the letters of a particular individual's genome that's within a price range that could be used as a routine clinical test. And we're going to hear in later talks from Professor King and from Professor Delante about how this kind of technology can lead to better diagnosis and in some rare but hopefully increasing cases, a better prognosis for a patient. This next slide is to try and summarize the current situation with clinical genetics for epilepsy. And And with pediatric cases of epilepsy, so these would be very severe early onset cases, sometimes with developmental delay and learning disability. We're at the stage now where approximately 20, perhaps even 30% of such cases can be explained by a particular underlying genetic mutation. And very often these changes are those de novo mutations that I was speaking about earlier. In the majority of these cases, unfortunately, the value is limited to just explaining that particular individual's epilepsy, why it's occurred. Now, that's obviously a very important thing in itself, but obviously as researchers and clinicians, the wish is to try and go further, and with that understanding of the underlying cause, hopefully be able to change treatment in a way that improves outcome for that individual. There are some examples of where understanding the mutation does allow a treatment that works really, really well for that particular cases, or that particular case, but right now that it's relatively limited, those examples. With adult epilepsy, and by adult epilepsy here, I mean that perhaps epilepsy that presents a little bit later on in life as opposed to in the first few weeks of life. Perhaps 5% of cases can be explained by one of these large underlying genetic changes, or if there's a very, very strong family history, a proportion of those cases can also be explained by a particular epilepsy gene. The other kind of recent progress that's been made with genetics of epilepsy is there are now tests that can be run that allow you to identify individuals who would be at risk of an adverse reaction to Tegretol or carbamazepine, which is a very commonly used epilepsy treatment. So in some patients, thankfully rarely, there can be a severe adverse reaction where there's a skin rash where a patient can end up, or an individual can end up in hospital. There are genetic tests now that can help identify those particular individuals, although there are challenges with getting those tests integrated into clinical care. Okay, so the kind of broad challenges for us as researchers to help with the treatment of epilepsy into the future, the first one I thought is to increase or improve our ability to identify the genetic cause of an individual's epilepsy. So what we mean here is improving the diagnostic yield. So if we were, say, to test the DNA sequence of 100 people with epilepsy, what's the proportion that we can explain? You know, is it 5%, 10%, and so on? So obviously the higher that value, in theory, the better, because it's explaining more of what the underlying problem is. But then once you've actually identified the underlying problem, the other major challenge is making a difference to that individual's care. So, you know, the dream is to be able to, well, ultimately cure that individual, but if not, at least identify a treatment quickly to which that patient will respond. And there are, and we'll hear later on tonight, some limited but remarkable examples of that actually being the case, where the exact treatment is identified. And I think the really exciting thing is sometimes it's a treatment that wasn't even realized would be effective for epilepsy. It could be something for something completely different. So in the context of the improved ability to diagnose epilepsy at a genetic level, there's been an initiative started by the Cure Foundation in the States called the Epilepsy Genetics Initiative. And we were hoping that Tracy Dixon Salazar could have been here with us tonight because she is leading this particular initiative. But the basic idea is for people with epilepsy who have one of these genomic genetic tests done, that they have the option to put that information into a database that allows researchers to better identify the genetic cause, either in that individual or other people with epilepsy who've had a sequence done. And the reason why sharing the information in this way is important is because our ability to actually identify a genetic cause is heavily dependent on the number of genetic sequences that we're able to read. So you can recognize what's actually causing someone's epilepsy from a different mutation that has no influence whatsoever on an individual's seizures. So we'll speak a little bit more about this later on. Just finally, in terms about sequencing here in Ireland, so we were lucky to get support from Science Foundation Ireland recently to get one of these sequencing machines installed at Beaumont Hospital. So we have been running, and indeed some of you might be participants in this particular study. We call it a biobank, where we're collecting blood and DNA from people with epilepsy. And we've been looking for genetic causes, either for the epilepsy in these individuals or for genetic factors that might guide treatment. And this work and this biobank has contributed to some of the discoveries that I alluded to earlier. But with this machine, what we're hoping to do now is start sequencing in more detail people who are participating in this biobank to see if we can identify genetic causes or things that might guide treatment in those individuals. So I'll just stress that at this point in time, this is regarded as a research tool, and by that we mean we don't bring the results into the clinical domain. It's purely for research purposes. Once the information is properly understood, then there is the potential to try and use this technology in the clinical domain. But right now at Beaumont Hospital, it's very much a research tool, but we're hoping that it can become diagnostic in the medium term. Okay, so just to summarize, all human populations are full of genetic variation, so it's important to realize this term mutation that you hear about in the media, that we are all full of mutations ourselves. Sometimes this variation can lead to seizure conditions in epilepsy. Our ability to identify these changes is increasing, and that's because of that new technology we call next-generation sequencing. It's critical that research is conducted to increase the value of this testing. So for us to be able to understand what the information means in the context of epilepsy, research needs to be done to improve that understanding. And just finally to say that Ireland, through various different speakers that are in fact all the speakers that are speaking tonight, are contributing to this research effort on a global level. And we'd like to also thank your participation in these studies, because that's obviously a critical aspect of it. So I'll stop there.